Welcome, dedicated learners, to the Deep Dive. Today we're embarking on a crucial journey into one of the most fundamental chapters in biology, the animal kingdom. Just look around. I mean, from the tiniest amoeba to the colossal whale, life bursts with incredible forms. Over 1 million animal species have been described. So how do we systematically unravel this immense diversity? How do we truly understand their relationships, their evolution, their unique adaptations? This deep dive will provide you with that essential roadmap. It's about laying a robust foundation for your Class 11 and 12 board exams and, most importantly, competitive exams like NEET. We have our expert guide here to lead us through this intricate classification.
Thank you. Yes, our mission today is precisely that, to systematically unravel the complexities of animal classification. We'll focus on the fundamental features that group animals together, then meticulously examine the distinct characteristics of each major phylum and class. Understanding this chapter conceptually is paramount. It's not just about, rote memorization. It's about building a strong foundation for higher biology, which in turn helps you secure those crucial marks in your exams. So let's unpack this together step by step.
Okay, let's start right at the beginning then. The basis of classification. Why do we even need to classify animals?
Right. Well, think about it. Over 1 million species described. Imagine the chaos without a system. Classification is absolutely essential for systematic study. It helps us organize this vast biological information into a logical, hierarchical structure.
Like organizing a massive library, maybe. Without a system, finding a specific book would be impossible.
Exactly like that. In biology, this systematic approach helps us understand evolutionary pathways, identify relationships between different groups, and practically, well, it lets us assign a proper, unique position to any newly discovered species. And for your exams, understanding why we classify is really just as important as how we classify.
Okay, got it. So what are these fundamental features we use? Despite all the diversity, there must be some common ground.
There is. And these fundamental features act as pillars for their classification. These are the core criteria we use. We'll be looking at things like how cells are arranged, the animal's body symmetry, the nature of their body cavity, or coelom as it's called, and also patterns we observe in their digestive, circulatory, and reproductive systems.
So each feature gives us a clue, distinguishing one group from another.
Precisely, and you need to pay close attention to these as they often form the basis of direct questions in NEET and the board exams.
Makes sense. Let's dive into the first one then, levels of organization.
Okay, so while all members of kingdom Animalia are multicellular, how their cells are organized varies significantly, and this represents increasing complexity. At the most primitive end, we have the cellular level of organization. You see this in animals like sponges, Phylum Porifera. Here, cells are basically arranged as loose aggregates.
So like individual workers doing specific jobs, but not really organized into teams.
That's a good analogy. There's some division of labor, sure, but no true tissues are formed. It's quite basic.
Okay. What's the next step up?
That would be the tissue level of organization. This is seen in Coelenterates, Phylum Sandaria. Here, cells that perform the same function actually group together to form proper tissues. It's a more coordinated and, more complex arrangement.
Right, from loose cells to organized tissues, and then
we get to the organ level of organization. Platyhelminthes, the flatworms, and higher phyla show this. Now, tissues are further organized into specialized organs. So, for instance, you might see a distinct excretory organ or reproductive organ, each made of different tissues working together for a specific function.
Okay, tissues combining into organs. And the highest level.
That's the organ system level of organization. This is what you see in annelids, arthropods, mollusks, echinoderms, and chordates, basically. Most of the animals we're familiar with. Here, multiple organs associate to form functional systems, each dedicated to a specific physiological process, like the digestive system or the circulatory system.
Allowing for really specialized and efficient processes.
Exactly. Highly specialized and efficient. And within these levels, we see different patterns in these organ systems. Let's quickly look at digestion system patterns. There are two main types. First, the incomplete digestive system. You find this in simpler forms like Platyhelminthes. It has just a single opening that serves as both mouth and anus. So food goes in, waste comes out, all through the same hole.
Okay. Not the most efficient setup, I guess.
Not really. In contrast, you have the complete digestive system. This is found in Aschelminthes, the roundworms, and all higher phyla. The system has two separate openings, a distinct mouth and a distinct anus.
Like a one-way street for food.
Precisely. It allows for unidirectional food flow, enabling much more specialized and efficient digestion as food moves through different parts of the gut.
Okay. And what about circulatory systems? You mentioned patterns there too.
Yes. We see two main circulatory system patterns. There's the open type. This is common in arthropods, for instance. Here, blood is pumped out of the heart, and it directly bathes the cells and tissues in open spaces or sinuses.
So the blood isn't always contained in vessels.
Exactly. It kind of sloshes around the organs. Then you have the closed type. This is seen in annelids and chordates. That's much more controlled and efficient. Here, blood circulates exclusively through a series of vessels, arteries, veins, and capillaries. This ensures more precise and efficient transport of nutrients, gases, and waste.
That's a really clear progression, definitely. From simple cellular aggregates to these complex organ systems. Could you help us understand why this increase in complexity is so significant from an evolutionary perspective? Especially, thinking about efficiency. What does an organ system level allow an animal to do that a tissue level animal just can't, or maybe not as well?
That's an excellent question and really vital for conceptual understanding. This increase in organizational complexity represents a major evolutionary leap. Think about sponges again. Coordination is limited, right? Each cell functions somewhat independently. Now jump to the organ system level. Organs associating into functional systems allow for highly specialized and efficient physiological processes. For instance, that complete digestive system means continuous unidirectional food processing. Different parts of the gut can specialize, some for breaking down food, others for absorbing nutrients. Much more efficient than an incomplete system where digestion and waste removal interfere.
Okay, specialization is key.
Exactly. And similarly, a closed circulatory system ensures blood is delivered precisely where it's needed. It allows for better regulation of flow and pressure, which is critical for supporting a larger, more active body. This specialization and coordination, they optimize energy use and maximize functional capacity. This enables animals to achieve greater metabolic rates, perform more complex behaviors, and ultimately adapt to a wider range of environments. That's what led to the incredible diversity we see today.
Right. It allows for a bigger, faster, and more complex life. Makes sense. Let's move on to the next fundamental feature you mentioned. Symmetry.
Yes, symmetry. This refers to the arrangement of body parts around a central axis or plane. It tells us a lot about an animal's lifestyle and how it interacts with its environment. First, we have asymmetrical animals. Sponges are mostly asymmetrical. Basically, no plane passing through their center can divide them into two equal halves.
Like trying to cut a natural sea sponge into two perfectly identical pieces is pretty much impossible, right? Because there's no set pattern.
Exactly. This lack of symmetry fits well with their sessile filter-feeding lifestyle. They don't really need a front or back.
Okay, then what?
Next is radial symmetry. An organism has radial symmetry when any plane passing through its central axis divides the body into two identical halves.
Think of a pizza or maybe a bicycle wheel.
Perfect examples. This is characteristic of coelenterates like jellyfish, Ctenophores, or comb jellies, and importantly, adult echinoderms, think starfish. This symmetry is often associated with sessile or slow-moving aquatic lifestyles. It allows them to sense stimuli or food coming from all directions equally.
No need for a defined front or back if you're just sitting there or drifting.
Precisely. And finally, we have bilateral symmetry. Here, the body can be divided into identical left and right halves by only one specific plane passing through the body.
Like us humans or an earthworm. If you draw a line down the middle, you get two mirror images.
Correct. This is observed in annelids, arthropods, and most higher animals. And this type of symmetry is a major evolutionary advancement because it promotes cephalization.
Cephalization. That's the development of a head region.
Exactly. The development of a distinct head region where sensory organs and nerve tissue are concentrated. This concentration allows for directed movement moving forward with purpose. It's highly advantageous for actively searching for food, escaping predators, or exploring new environments.
So bilateral symmetry allows for more directed movement and the development of a head, while radial symmetry offers that multi-directional sensing. Now, is there a specific phylum where we see a really fascinating shift or maybe an interesting exception regarding symmetry that our students should be particularly aware of for exams? I'm thinking perhaps echinoderms.
Ah, yes. That's a crucial point for competitive exams, especially NEET. You've hit on a very important exception. While adult echinoderms, like starfish or sea urchins, clearly show radial symmetry, their larval stages are actually bilaterally symmetrical.
Oh, wow. So they start bilateral and become radial.
Yes. This unique paradox strongly indicates their evolutionary history. It suggests echinoderms evolved from bilaterally symmetrical ancestors, and the radial symmetry in adults is a secondary adaptation, likely linked back to their sessile or slow-moving bottom-dwelling lifestyle as adults.
That's a classic exam trap, then.
Definitely. For exam purposes, remember, adult echinoderms are radially symmetrical, but their larvae are bilaterally symmetrical. Very common trick question.
Good to know. Okay, moving on to another critical classification feature, diploblastic and triploblastic organization. Sounds complex.
It sounds complex, but the concept is straightforward. This classification is based on the number of embryonic germ layers. These are the fundamental layers of cells formed during early development, from which all body tissues and organs ultimately differentiate. Think of them as the basic blueprints for the animal body. So first we have diploblastic animals. These animals possess two primary embryonic layers, an external layer called the ectoderm and an internal layer called the endoderm. What's interesting here is that in between these two layers, there's often an undifferentiated, sort of non-cellular, jelly-like layer called the mesoglea.
Okay, two main layers plus a sort of middle jelly.
Kind of. Coelenterates like hydra and jellyfish are the classic examples. The ectoderm typically gives rise to the outer coverings and the nervous system, while the endoderm forms the lining of the digestive tract and associated glands.
Right, and triploblastic, that must mean 3 layers.
Exactly. Triploblastic animals have a third distinct germinal layer called the mesoderm, and it's situated precisely between the ectoderm and the endoderm. This includes all phyla, from Platyhelminthes, the flatworms, all the way up to chordates, including us.
So this mesoderm must be pretty important.
It's a monumental evolutionary step. Why? Because the mesoderm gives rise to a huge array of complex structures crucial for higher life forms. This includes muscles, connective tissues, the circulatory system, the excretory system, and most other internal organs. The presence of this third germ layer allows for significantly greater complexity and the development of specialized organ systems.
Okay, that makes sense. For students preparing for NEET, understanding the origin of different organs from these germ layers is absolutely crucial. Could you briefly elaborate on what some key structures arise from each of these 3 embryonic layers? Like, where does our nervous system come from, or our digestive lining?
Absolutely. This is a high-yield area for exams. Let's break it down simply. From the ectoderm, the outermost layer, you primarily get structures that interact with the outside world. Think of your skin, the epidermis, hair, nails, glands in the skin. Crucially, the entire nervous system, brain, spinal cord, all peripheral nerves develops from the ectoderm. Also, sensory organs like the eyes and inner ear and the lining of the mouth and nasal cavity originate here.
So outer stuff and the control system. Got it. What about the inside layer, the endoderm?
The endoderm, the innermost layer, primarily forms the lining of the internal body systems, most notably the entire alimentary canal or digestive tract from the pharynx down to the rectum, along with associated glands like the liver and pancreas are derived from the endoderm. Also, the lining of the respiratory system, including the lungs and trachea, originates from this layer. Think of it as lining the inner tube of the body.
Okay, inner linings. And the middle layer, the mesoderm. That sounds like it does a lot.
It really does. The mesoderm, the middle layer, is where the bulk of the sort of working body structures come from. This layer allows for the greatest complexity. All types of muscles develop from the mesoderm. The circulatory system, heart, blood vessels, blood cells, is mesodermal. The excretory system, like kidneys and ureters, the reproductive system, gonads, ducts, and the entire skeleton, including bones and cartilage, also arise from the mesoderm. Even connective tissues like cartilage, bone, fat, and the dermis layer of the skin are all mesodermal in origin. So yes, the mesoderm truly enables the development of complex movement, internal transport, and regulatory systems.
Wow, okay. Ecto, endo, meso. Crucial distinctions. What's our next fundamental feature? I think you mentioned the coelom.
Yes, the coelom. The coelom is basically the body cavity. Specifically, it's the fluid-filled space located between the body wall and the gut wall. Its presence, absence, or the way it's formed is a very important criterion for classifying animals.
Okay, so how do we categorize animals based on this coelom?
Right, we can group them into 3 main categories. First, coelomates. These are animals possessing a true coelom. What makes it true? It's a body cavity properly lined on all sides by mesoderm. This lining ensures the organs within the coelom are well supported and can even move independently.
And which animals are coelomates?
This includes annelids, mollusks, arthropods, echinoderms, hemichordates, and all chordates. A true coelom provides space for organ development, offers protection, and can act as a hydrostatic skeleton aiding movement.
Okay, true coelom means lined by mesoderm. What's next?
Second, we have pseudocoelomates. Here, a body cavity is present, but it's not truly lined by mesoderm. Instead, the mesoderm is present as scattered pouches or isolated bits of tissue between the ectoderm and endoderm. It doesn't form a continuous lining.
Ah, so it's a cavity but not properly lined, hence pseudo or false.
Exactly. Aschelminthes, the roundworms, are the classic and most important example of pseudocoelomates for your exams. This false coelomate still provides some space for organs and acts as a hydrostatic skeleton to some extent, but it lacks the full structural support and organizational advantages of a true coelom.
Okay, Aschelminthes equals pseudocoelomates, and the third type.
Finally, there are acoelomates. These are animals in which the body cavity is completely absent. Their body is essentially solid between the outer wall and the gut. There's no internal fluid-filled space there. Platyhelminthes, the flatworms, fall into this category.
So no cavity at all. Flatworms are flat for a reason, maybe helps with diffusion.
Precisely. Their flattened body shape helps in nutrient and waste diffusion in the absence of a body cavity and circulatory system.
Okay, so the coelom is clearly more than just an empty space. It plays roles in organ protection, allows for independent organ movement, and even functions as a hydrostatic skeleton. For exam questions, what's a common misconception students have about the pseudocoelom that we should clarify, make sure they don't get tricked?
That's a great point, because the distinction between pseudocoelom and true coelom is a classic area of confusion. I think the most common misconception is that students think it's just a partially developed true coelom, but the crucial difference lies in its lining. Remember, in a true coelomate, the cavity is fully lined by mesoderm. This allows for peritoneal membranes that suspend organs. In a pseudocoelomate, the mesoderm does not form a continuous lining, it's scattered. The cavity itself is essentially an unlined space between the endoderm and the mesoderm-ectoderm layers.
So the pseudo really refers to the lack of that complete mesodermal lining.
Exactly. That's the defining characteristic for Aschelminthes. For your exams, when you think pseudocoelomate, immediately think Aschelminthes or roundworms. This clearly sets them apart from both acoelomates, no cavity, and true coelomates, fully lined cavity.
Got it. No mesodermal lining pseudo. Okay, next up, segmentation.
Yes, segmentation. In some animals, the body is externally and internally divided into a series of repeated units or segments. This pattern is known as metameric segmentation, and the phenomenon itself is called metamerism.
Like in an earthworm. You can clearly see the rings.
The earthworm, an annelid, is the classic example. If you look closely, you see the ring-like segments. And within these segments, there's often a serial repetition of at least some organs like nerves, blood vessels, excretory organs. Segmentation allows for specialized functions in different segments and provides a significant advantage for efficient locomotion, especially for things like burrowing or crawling.
This looks like a really great evolutionary leap. How does metamerism, this repetition, contribute to a more advanced body plan compared to unsegmented forms? What specific advantages does it provide?
The development of metamerism is indeed a significant evolutionary advancement. Compared to unsegmented forms, it offers several key advantages. Firstly, it allows for specialization of body regions. Different segments or groups of fused segments, called tegmata, can become modified for specific functions. Think about insects' head for sensing and feeding, thorax for locomotion, abdomen for digestion and reproduction. That leads to greater efficiency.
Right, division of labor between body parts.
Exactly. Secondly, segmentation provides increased flexibility and mobility. Each segment can move somewhat independently, controlled by its own muscles, allowing for more complex and precise movements. Think of the wave-like motion of an earthworm burrowing, highly effective.
More control.
Yes. Thirdly, it offers redundancy and resilience. If one segment is injured, the others can often continue to function, providing a degree of protection and a better chance of survival. And finally, it provides a more efficient basis for the development of a hydrostatic skeleton in coelomate segmented animals, allowing for powerful and localized shape changes that aid in locomotion.
So it's not just simple repetition. It opens the door for specialization, better movement, and backup systems.
Precisely. It's a very effective body plan.
Okay. And the final fundamental feature you listed was the notochord.
Yes, the notochord. This is a mesodermally derived, flexible, rod-like structure. It forms on the dorsal side, the backside of the body, during embryonic development in some animals. Its presence or absence is a primary distinguishing feature that divides the entire animal kingdom into two major groups.
Chordates and non-chordates, right?
Correct. Animals possessing a notochord at some stage of their life are called chordates. This includes a vast and diverse group, from simple tunicates all the way to fish, amphibians, reptiles, birds, and mammals. Conversely, animals that do not form this structure at any stage are called non-chordates. This includes all the phyla we've discussed so far, from Porifera up to Echinodermata. The notochord provides skeletal support and serves as an axis around which the vertebral column later develops in vertebrates. It's a foundational structure for the body plan of all chordates.
This notochord sounds like a very foundational structure indeed. Does its presence signify a higher level of organization or maybe a greater potential for complex development compared to animals without it? What's the evolutionary implication here?
That's a very insightful observation. The notochord does indeed signify a significant evolutionary advancement and a higher potential for complex development. It provides the first real axial support to the embryonic body. This rigid yet flexible axis allows muscles to contract more efficiently against it, enabling better locomotion, especially swimming movements. More importantly, perhaps, it acts as a crucial signaling center during embryonic development. It influences the formation of the nervous system above it and the vertebral column around it later on.
And so it guides development.
Yes, it plays a critical inductive role. In vertebrates, the notochord is later replaced by the vertebral column, which offers even stronger, more complex skeletal support, allowing for larger body sizes and more sophisticated movement. So while the notochord itself might seem relatively simple, it represents a pivotal innovation. It paved the way for the evolution of the highly complex, segmented vertebral column and the diverse, active lifestyles we see in vertebrates. You can think of it as the evolutionary precursor to the backbone.
Okay, that clarifies its importance. So now that we have the basic criteria levels of organization, symmetry, germ layers, coelom, segmentation, notochord, let's dive into the actual phyla.
Exactly. Let's go through the major phyla, from the simplest to the most complex. And for your exams, it's absolutely crucial to pay close attention to the unique features and representative examples for each. These are very frequently tested. Our first stop is Phylum Porifera. These are commonly known as sponges. They are mostly marine, largely asymmetrical, and primitive multicellular animals. They have that very basic cellular level of organization. Their cells are just loose aggregates, remember? No true tissues.
Right, the simplest multicellular setup. What's their defining feature?
A truly distinguishing feature is their water transport or canal system. Water enters through countless minute pores called ostia, flows into a central cavity called the spongocoel, and then exits through a larger opening, the osculum. This pathway is absolutely crucial for everything. Food gathering by filtering particles from the water, respiratory exchange as oxygen diffuses in, and waste removal as waste products are carried out.
Like a built-in plumbing and feeding system.
Exactly. Imagine water constantly flowing through a sieve, bringing in nutrients and carrying away waste. And lining the spongicoel and canals are specialized flagellated cells called choanocytes or collar cells. These cells, with their beating flagella, create the water current and, importantly, filter out the food particles. Digestion in sponges is entirely intracellular, happens within individual cells. Their body gets support from a skeleton made of tiny spicules, microscopic needle-like structures made of calcium carbonate or silica, or flexible spongin fibers, a network of protein.
Okay, and how do they reproduce?
Sponges are generally hermaphrodite, meaning the same individual produces both eggs and sperms, though usually not at the same time. They reproduce asexually by fragmentation. A piece breaks off and grows into a new sponge. And they also reproduce sexually by gamete formation. Fertilization is internal, and development is indirect, meaning there's a distinct larval stage that looks different from the adult.
Any key examples we should remember?
Definitely. Remember Cycon, also called Cypha, Spongilla, this one's important as it's a freshwater sponge, an exception, and Euspongia, the common bath sponge.
That water canal system is quite unique. How does this system, despite its cellular level simplicity, allow sponges to be so successful? They've been around for ages, right? How do they thrive being so, well, simple?
That's a perceptive question. Despite their cellular level, the water canal system is absolutely fundamental to sponges' evolutionary success and persistence. Its primary benefit is highly efficient filter feeding. The continuous water flow through thousands of ostia, driven by those choanocytes, brings a constant supply of microscopic food particles directly to the cells that can phagocytose them. This passive feeding strategy is very energy efficient for a sessile animal.
So food just comes to them.
Pretty much. Secondly, it's a multi-purpose utility system. Beyond nutrition, it ensures efficient respiratory exchange, getting oxygen in and CO2 out, and provides an effective mechanism for waste removal. Everything relies on that water flow. Lastly, it even plays a role in reproduction. Sperm released by one sponge can be carried by water currents into another for internal fertilization. The larvae are also dispersed by water currents. So this single, relatively simple system integrates multiple vital physiological functions. It allows them to effectively extract resources from their environment while living a low-energy, sessile existence. That's why they've been so successful and persisted for hundreds of millions of years.
Amazing efficiency from simplicity.
Okay.
Okay, next phylum, Coelenterata or Cnidaria?
Yes. Phylum Coelenterata, also widely known now as Cnidaria. These are aquatic, predominantly marine animals. They can be either sessile, like sea anemones or corals, or free-swimming, like jellyfish. They are radially symmetrical and diploblastic, remember, 2 germ layers, and they have the tissue level of organization.
And a most distinctive feature.
The stinging cells.
Right.
Exactly. Their most distinctive feature, and where the name Cnidaria comes from, is the presence of specialized stinging cells called cnidoblasts, or cnidocytes. These are found all over their tentacles and body. Each cnidoblast contains a complex intracellular structure called a stinging capsule or nematocyst. Think of these as tiny, coiled, venomous harpoons that can be rapidly discharged upon stimulation.
Harpoons, what do they use them for?
Primarily for anchorage, defense against predators, and crucially for capture of prey. They can paralyze or ensnare small animals. Cnidarians possess a central gastrovascular cavity, a combined digestive and circulatory space, with just a single opening that functions as both mouth and anus, located on a structure called the hypostome. Digestion is both extracellular, partially broken down in the cavity, and intracellular, completed inside cells lining the cavity. Some cnidarians, notably corals, secrete a hard external or internal skeleton composed of calcium carbonate, forming coral reefs.
Okay, and you mentioned different body forms.
Yes, cnidarians exhibit 2 basic body forms. There's the polyp, which is typically sessile, cylindrical, like Hydra or Adamsia, sea anemone. And there's the medusa, which is umbrella-shaped and free-swimming, like Aurelia, jellyfish.
Polyp and Medusa, like 2 different lifestyles.
Exactly. And here's where it gets really interesting and important for competitive exams. Some cnidarians, like Obelia, exhibit metagenesis. This is also known as alternation of generations. This means they alternate between these 2 body forms in their life cycle. Typically, polyps produce medusa asexually, by budding, and then the medusa reproduce sexually, producing gametes, which develop into polyps again.
Asexual polyps making sexual medusae, which make polyps. That's a cycle.
Precisely. This unique life cycle pattern is a very frequently tested concept. Remember Obelia as the key example.
Got it. Any other important examples?
Yes. Remember Phacelia, the Portuguese man-of-war, which is actually a colony, Adamsia, sea anemone, and Aurelia, jellyfish.
The concept of metagenesis can be a bit tricky for students. Could you simplify that cycle of polyp to medusa and back again, and maybe highlight why this unique life cycle is evolutionarily advantageous for cnidarians like Obelia?
You're right. Metagenesis is a sophisticated strategy, but we can simplify it. Let's use Obelia as our example. The polyp stage is typically sessile, anchored to a surface, often forming colonies. These polyps are focused on feeding and growth. They reproduce asexually by budding off tiny, free-swimming medusae.
Okay, so the stationary polyps release mobile medusae.
Exactly. These medusae are the dispersal stage. They float or swim away, carrying the genes to new locations. The medusae are the sexual stage. They produce eggs and sperm, which are released into the water. After fertilization, a zygote develops into a swimming larva, planula larva. This larva eventually settles down on a suitable surface and metamorphoses into a new polyp, starting the cycle again.
So asexual reproduction for local growth, sexual reproduction for dispersal and genetic mix.
That's the essence of it. It offers the best of both worlds. The polyp stage is excellent for exploiting local resources and rapid asexual proliferation when conditions are good and stable. The medusa stage, being mobile, provides crucial dispersal. This allows the species to colonize new habitats, escape unfavorable local conditions, and promote genetic mixing across a wider geographic range, which enhances genetic diversity and adaptability. It really maximizes their chances of survival and spreading throughout diverse marine environments.
Brilliant strategy. Okay, next up, phylum Ctenophora, sea walnuts.
Yes, Phylum Ctenophora, commonly called sea walnuts or comb jellies. These are exclusively marine, radially symmetrical, and diploblastic animals, also possessing the tissue level of organization. They look a bit like jellyfish, but are quite different.
How are they different? What's their defining feature?
Their most defining characteristic and how they move is the presence of 8 external rows of ciliated comb plates. These are rows of fused cilia that beat in a coordinated wave-like rhythm, propelling the ctenophore, usually mouth first, through the water. These comb plates are their primary means of locomotion. Digestion is also both extracellular and intracellular.
And they glow, right? Bioluminescence.
Yes. Another truly remarkable characteristic is bioluminescence. Many actinophores can produce light, often creating spectacular displays in the ocean depths. It's a very well-marked feature in this phylum. Reproduction is interesting too. Sexes are not separate, they are hermaphrodites, and reproduction occurs only by sexual means. There's no asexual reproduction typically. Fertilization is external, and development is indirect, usually involving a distinct larval stage.
And examples.
Common examples include Pleurobrachia and Scedoplana.
That bioluminescence is fascinating. How does this ability actually benefit the Ctenophores in their dark marine environment? Is it mainly for attracting prey, or is it more of a defense mechanism, or maybe both?
That's a great question, and the answer is likely multifaceted. Bioluminescence in ctenophores probably serves multiple ecological roles, and its function can vary between species. It's generally thought to function in defense. A sudden flash of light when disturbed might startle or confuse a potential predator, allowing the ctenophore to escape. Some suggest it might act as a burglar alarm. Lighting up might attract a larger predator that would then prey on the initial attacker.
A risky defense.
Possibly. In some cases, it might be used for prey attraction, perhaps luring smaller organisms towards them in the dark. It could also play a role in intraspecific communication, maybe related to mating, though that's less certain. So it's likely a versatile tool in their survival kit for defense, maybe offense, maybe communication. It certainly makes them one of the most visually stunning inhabitants of the ocean.
Very cool. Okay, now we move to Phylum Platyhelminthes, the flatworms.
Yes, Phylum Platyhelminthes, commonly known as flatworms. The name itself describes them perfectly. Their body is dorsoventrally flattened, meaning flattened from top to bottom, often like a ribbon or a leaf. While many flatworms are free-living, like Planaria, a significant and medically relevant group are the endoparasites, living inside the bodies of other animals, including humans.
And these represent a step up in complexity, right?
Absolutely. This phylum marks several significant evolutionary advancements. Flatworms are bilaterally symmetrical. They are the first phylum we encounter that is triploblastic, possessing all three embryonic germ layers, ectoderm, mesoderm, endoderm. However, they are also acoelomate animals, meaning they completely lack a true body cavity. Coelom. The space between the organs is filled with a type of connective tissue called parenchyma. They exhibit the organ level of organization.
And being parasites, they must have special adaptations.
They do. In parasitic forms like tapeworms and flukes, you often find specialized structures like hooks and suckers. These are used for firmly attaching to the tissues of their host. Many internal parasites also absorb nutrients directly from the host through their entire body surface, a very efficient way to feed when you're surrounded by digested food.
Clever, in a parasitic way. How do they handle waste and water balance?
They have specialized excretory cells called flame cells, or protonephridia. These are involved in both osmoregulation, water balance, and excretion of metabolic wastes. In terms of reproduction, sexes are generally not separate. Most flatworms are hermaphrodite, monoecious. Fertilization is internal, and development often involves complex life cycles with multiple larval stages, sometimes involving intermediate hosts. And some members, famously Planaria, possess an exceptionally high capacity for regeneration. You can cut them into pieces, and each piece can regrow into a whole new worm.
Incredible. Key examples.
Important examples, especially the parasitic ones, include Tenana, the tapeworm, and Fasciola, the liver fluke, and Planaria for regeneration.
The parasitic nature of many flatworms is a very significant aspect. Thinking about our students in India, what are some real-world implications or health concerns related to these parasites? Parasitic infections can still be a public health challenge there, right?
That's a vital connection to make, absolutely. In India, parasitic flatworm infections like teniasis, caused by Tinea species, and fascioliasis, caused by Fasciola, do pose significant public health concerns, particularly in areas with perhaps inadequate sanitation or specific dietary practices involving undercooked meat or contaminated water plants. For instance, Teniasis, which you get from consuming undercooked pork or beef containing tapeworm cysts, is bad enough in the gut. But if the larval stage, called cysticercosis, develops in humans, especially from the pork tapeworm, it can migrate to tissues like muscles, eyes, and even the brain.
The brain. That sounds serious.
It is. Neurocysticercosis is actually a major cause of acquired epilepsy in many parts of the world, including India. Similarly, fascioliasis, the liver fluke infection, typically acquired from eating raw aquatic plants like watercress contaminated with larval stages, causes significant liver damage, inflammation, jaundice, and abdominal pain. Both infections can lead to chronic illness, malnutrition, and reduced productivity. So understanding their biology and life cycles isn't just for scoring marks in NEET. It's crucial for public health awareness, prevention through proper cooking and sanitation, and control strategies.
A very important reminder of the real-world relevance. Okay, next phylum. Aschelminthes, the roundworm.
Yes, phylum Aschelminthes. If you were to cut one in cross-section, its body would be circular, hence the common name roundworms. They are incredibly abundant and diverse. You find them everywhere. They can be free-living in soil and water, both aquatic and terrestrial, or parasitic in plants and animals, including humans, where they cause many diseases.
And organizationally, where do they fit?
They exhibit the organ system level of body organization. They are bilaterally symmetrical and triploblastic. And uniquely, for your exams, remember this, they are pseudocoelomate animals. They have that false body cavity, not fully lined by mesoderm. This is a key distinguishing feature.
By the pseudocoelom. What about their gut?
A significant evolutionary step here is that they possess a complete alimentary canal. It has two separate openings, a mouth at one end and an anus at the other, and often a well-developed muscular pharynx for sucking in food. This is much more efficient than the single opening in flatworms. For excretion, they typically have an excretory tube or canal system that removes body waste from the body cavity through an excretory pore.
And sexes.
Unlike the hermaphroditic flatworms, in Aschelminthes, sexes are usually separate. They are dioecious, meaning there are distinct male and female individuals. Often, they exhibit sexual dimorphism where the female is noticeably larger or longer than the male. This is common in Ascaris, for example. Fertilization is internal. Development may be direct, where the young resemble miniature adults, or indirect, involving larval stages.
And important examples, especially the parasitic ones.
Yes, several medically important examples fall here. Ascaris, the common roundworm causing ascariasis, Wuchereria, the filaria worm causing elephantiasis, and Ancylostoma, the hookworm causing anemia.
The distinction between pseudocoelomates and acoelomates, and then true coelomates, is definitely a frequent point of confusion. Could you just quickly reiterate what makes the pseudocoelom pseudo, and why Aschelminthes are such key examples students absolutely must remember for their exams?
Yes, let's solidify this as it's critical. What makes the pseudocoelom pseudo is precisely the lack of a complete, continuous mesodermal lining surrounding the body cavity. Remember, in a true coelomate, the mesoderm splits, creating a space within the mesoderm itself, fully lined by it. In acoelomates like flatworms, there's no cavity at all, just solid tissue. In pseudocoelomates like Aschelminthes, there is a fluid-filled cavity between the gut, endoderm, and the body wall, ectoderm mesoderm, but this cavity is not lined by a layer of mesodermal cells. The mesoderm is present, but it doesn't form that complete peritoneal lining.
So it's an unlined space, essentially.
Exactly. It functions as a hydrostatic skeleton and allows some room for organs, but it's structurally and developmentally different from a true coelom. And Aschelminthes are the classic textbook example of pseudocoelomates. For your exams, this link, Aschelminthes pseudocoelomate roundworms, is absolutely fundamental. Memorize it. It clearly separates them from the acoelomate Platyhelminthes before them and the coelomate Phyla that come after.
Crystal clear. Aschelminthes and coelomates. Got it. Moving forward, we reach phylum Annelida, the segmented worms.
Yes, phylum Annelida. Think earthworms, leeches, and marine worms like Nereis. Annelids are quite adaptable, found in aquatic environments, both marine and freshwater, or in terrestrial soil. Many are free-living, but some are parasitic, like leeches. Organizationally, they take several steps forward. They exhibit the organ system level of body organization. They display clear bilateral symmetry. They're triploblastic. And crucially, they are the first phylum we've met that is truly metamerically segmented and properly coelomate animals. They possess a true coelom.
Metameric segmentation, that's the repetition of segments we talked about earlier.
Exactly. Their body surface is distinctly marked out externally into segments, or metameres, like the rings on an earthworm. And this segmentation is also reflected internally to some extent. This gives the phylum its name, from the Latin annulus, meaning little ring.
How do they move?
They possess well-developed longitudinal and circular muscles which work against their hydrostatic skeleton, the fluid-filled tubulum, to produce movement, like crawling and burrowing. Aquatic annelids, like Nereis, often have paired lateral appendages called parapodia on each segment. These are fleshy, paddle-like structures, often bearing bristles, setae, used for swimming and also sometimes for respiration.
And their internal systems. Yeah. Circulation.
Annelids show another major evolutionary advancement. They possess a closed circulatory system. This means blood circulates entirely within a network of blood vessels, ensuring more efficient and directed transport of oxygen and nutrients compared to the open systems we saw earlier. For excretion and osmoregulation, they have specialized excretory organs called nephridia, singular, nephridium. Typically, there's a pair of these coiled tubules in each body segment, filtering waste from the coelomic fluid and blood.
What about their nervous system?
Their nervous system is also well developed. It usually consists of a pair of cerebral ganglia, a simple brain, connected by nerves to a double ventral nerve cord that runs the length of the body, with ganglia and lateral nerves in each segment.
And reproduction?
It varies. Neri-Hi, the marine worm, is dioecious, separate sexes. However, earthworms, Pheretima, and leeches, Hirudinaria, are monoecious, hermaphrodites, although they usually cross-fertilize. Reproduction is primarily sexual.
Okay, key examples again.
Definitely remember Nereis, the clamworm or sandworm, Pheretima, the common Indian earthworm, and Hirudinaria, the blood-sucking leech.
The presence of a true coelom and that distinct metameric segmentation in annelids marks a major evolutionary step up. How do these two specific features working together really contribute to the success and diversity of annelids? How do they enable more complex behaviors and let them occupy different ecological roles?
That's a great question because it highlights the power of combining these evolutionary innovations. The true coelom in annelids, being fully lined by mesoderm, functions very effectively as a hydrostatic skeleton. Think about it. The fluid inside the coelom is incompressible. When the circular and longitudinal muscles contract against this fluid in a specific segment, they can change the shape of that segment, making it longer and thinner or shorter and fatter.
Like how an earthworm moves.
Exactly. Each coelomic compartment within a segment can be pressurized somewhat independently. This allows for very precise and localized changes in body shape. The characteristic peristaltic movement of an earthworm, where waves of contraction pass down the body, is incredibly efficient for burrowing through soil, something unsegmented or pseudocoelomate animals really can't do as effectively. Now, combine that with metameric segmentation. This repetition allows for specialization. While segments might look similar externally, internally they can become specialized. More importantly, it allows for independent control of segments, leading to more sophisticated locomotion.
And redundancy too, right?
Yes, redundancy is a benefit too. Damage to one segment might not be fatal. Furthermore, this segmentation provides multiple points for muscle attachment, allowing for stronger and more controlled movements. The closed circulatory system efficiently supports this increased activity by delivering oxygen and nutrients effectively throughout the segmented body. So the true coelom provides the mechanical basis for efficient hydrostatic movement, and segmentation allows for regional specialization and refined control. Together, these features opened up new ecological possibilities, like burrowing deep into sediment or soil, leading to the diversity we see in annelids today.
Makes perfect sense. Coelom plus segmentation, better movement and specialization. Okay, brace yourselves, everyone. Next is the big one, Phylum Arthropoda, the largest phylum.
Indeed. Phylum Arthropoda is truly astounding in its diversity and numbers. This is, without question, the largest phylum of Animalia. Insects alone make up a huge portion of this. Over 2/3 of all named species on Earth are arthropods. Organizationally, they are at the organ system level. They are bilaterally symmetrical, triploblastic, clearly segmented, and coelomate animals.
What's their absolute defining characteristic? The exoskeleton.
That's definitely one of the most defining features. Their body is covered by a hard, protective, non-living, chitinous exoskeleton. This rigid outer covering provides excellent structural support, muscle attachment points, and crucially, protection against predators and water loss, which was key to conquering land.
But doesn't a rigid exoskeleton make growth difficult?
It does. That's why arthropods must periodically shed their exoskeleton in a process called molting or ecdysis to allow for growth. They're quite vulnerable during this period. Their body is typically divided into distinct regions or tagmata, usually head, thorax, and abdomen, although fusion of these regions can occur in some groups, like cephalothorax in spiders and crustaceans.
And the name Arthropoda itself, jointed legs.
Precisely. The very name Arthropoda means jointed appendages, from Greek arthros, joint, poda, foot appendage. This is perhaps their most distinctive feature. They possess paired, jointed limbs that are incredibly versatile. These appendages have been modified through evolution for a vast array of functions: walking, jumping, swimming, feeding, jaws, maxillae, sensing antenna, defense claws, respiration, and even reproduction. This adaptability of appendages is a huge reason for their success.
Wow. What about breathing? They live everywhere.
They show remarkable diversity in respiratory organs adapted to their environment. Aquatic forms often have gills, like crustaceans, or book gills, like horseshoe crabs. Terrestrial forms might have book lungs, like spiders and scorpions, or a highly efficient network of air tubes called a tracheal system, characteristic of insects, millipedes, centipedes, that delivers oxygen directly to the tissues.
And circular.
Their circulatory system is of the open type. A dorsal heart pumps blood, hemolymph, into arteries, which then empties into open spaces or sinuses, collectively called the hemocole, directly bathing the tissues before returning to the heart.
How about senses and waste removal?
They possess well-developed sensory organs, including antennae for touch and smell, compound eyes made of many individual units, great for detecting movement, and simple eyes, ocelli, and statocysts, balancing organs. For excretion, terrestrial arthropods, especially insects and myriapods, primarily use Malpighian tubules. These tubules absorb waste from the hemolymph and pass them into the hindgut for elimination. Aquatic forms often use different structures, like green glands or coxal glands.
Reproduction.
They're mostly dioecious, separate sexes. Fertilization is usually internal. They are mostly oviparous, egg-laying, although some are oviviparous or oviparous. Development may be direct or, very commonly, indirect, involving larval stages and metamorphosis, like caterpillar to butterfly.
Examples are countless, but maybe some key ones.
Let's mention a few categories. Economically important insects, apis, honeybee, bombyx, silkworm, laxifer, legless insect, vectors of disease, anopheles, culex, Aedes, all mosquitoes, gregarious pest, locusta, locust, a fascinating living fossil, limulus, king crab, and then spiders, scorpions, crabs, prawns, butterflies, beetles. The list is enormous.
Given their incredible diversity and sheer numbers, this is a classic exam question.
Ugh.
What are the primary reasons for the evolutionary success of arthropods? Is it just the exoskeleton and jointed legs, or is it a combination of factors that allowed them to absolutely dominate the animal kingdom?
That is indeed a very popular and important question. While the chitinous exoskeleton and jointed appendages are undeniably crucial pillars of their success, it's really the powerful combination of these features along with several other key innovations that propelled them to such dominance. The exoskeleton, as we said, provides support, protection, and vital prevention of desiccation, water loss, which was absolutely critical for colonizing terrestrial environments. It allows them to live in dry places where many other invertebrates can't. The jointed appendages are revolutionary in their versatility. They can be highly modified for specialized functions: walking, running, jumping, flying an insect, digging, grasping, manipulating food, sensing the environment. This adaptability allows them to exploit an incredible variety of ecological niches and food sources.
So exoskeleton for protection and land life, legs for doing everything else.
Exactly, but there's more. Segmentation and tegumentization, fusion of segments into specialized body regions like head, thorax, abdomen, allow for increased functional specialization and efficiency. Many arthropods, especially insects, have high reproductive rates and undergo metamorphosis. Metamorphosis is brilliant because the larval and adult stages often utilize different food sources and habitats, reducing competition within the species. Their diverse and highly efficient respiratory systems, like the tracheal system in insects, allow for effective gas exchange to support the high metabolic rates needed for activities like flight. And finally, their well-developed sensory organs, like compound eyes and antennae, enable them to effectively perceive and interact with their environment, find food, detect mates, and avoid predators. So it's not just one thing, it's the remarkable integration of the exoskeleton, versatile jointed appendages, segmentation, efficient respiration, metamorphosis, high reproduction, and advanced sensory systems that has allowed arthropods to successfully colonize nearly every habitat on Earth, leading to their unparalleled numbers and diversity.
A winning combination indeed. Okay, after the biggest phylum, what's next? Phylum Mollusca.
Yes, Phylum Mollusca. This is the second largest animal phylum after Arthropoda. Mollusks are also incredibly diverse. They are found in terrestrial environments like snails and slugs, or aquatic habitats, both marine and freshwater. They exhibit the organ system level of organization, are bilaterally symmetrical, though some, like snails, become asymmetrical due to torsion, triploblastic, and coelomate animals, though the coelom is often reduced.
What are their key features? They often have shells, right?
Yes. A common feature is that their body is typically covered by a calcareous shell secreted by the mantle. This shell can be external, like in snails or clams, internal, like in squid or cuttlefish, or completely absent, like in slugs or octopus. Their body is generally unsegmented, unlike annelids or arthropods, and typically has three distinct parts. A distinct head region, which may be reduced in some, usually bearing sensory organs, a large ventral muscular foot, primarily used for locomotion, and a visceral hump or mass dorsally, which contains most of the internal organs.
You mentioned the mantle secreting the shell. Tell us more about the mantle.
Right. A characteristic feature is a soft, slushy layer of skin called the mantle, or pallium, which covers the visceral hump. As you said, its outer surface secretes the shell. The mantle also encloses a space between itself and the visceral hump called the mantle cavity. This cavity is important because it usually houses feather-like gills, pectinidia, which are responsible for respiration. The mantle cavity is also where excretory products and gametes are released.
So the mantle cavity is like a multi-purpose chamber. What about feeding?
The anterior head region typically has sensory tentacles, and the mouth of most mollusks, except bivalves, contains a unique characteristic feeding organ called the radula.
Radula, what's that?
The radula is like a minutely toothed tectinous ribbon stretched over a cartilaginous support. It's used like a file or a rasp to scrape algae off rocks, tear up food, or even drill holes in other shells. It's a very distinctive molluscan feature. In terms of reproduction, mollusks are usually dioecious, separate sexes, though some are hermaphrodites, and mostly oviparous, egg-laying. Development is often indirect, involving one or more larval stages like the trochophore or veliger larva.
And examples?
Many familiar ones: Phyla, the apple snail; Punctata, the pearl oyster; Sepia, cuttlefish; Loligo, squid; octopus, devilfish; Aplisiae, sea hare; Dentalium, tusk shell; and Chaetoplera, chitin.
The radula for feeding and the mantle are such defining characteristics. You explained the mantle cavity's role, but how does the mantle itself, the tissue layer, contribute to the overall physiology of a mollusk? Does it do more than just form the cavity and secrete the shell?
That's a good point to clarify. The mantle tissue itself is indeed more than just a passive covering or shell-secreting layer. It's a dynamic and physiologically active structure. Besides secreting the shell, its surface often plays a role in gas exchange, supplementing the gills, especially in terrestrial snails or aquatic species where gills might be reduced. The mantle edge is often equipped with sensory receptors, allowing the mollusk to sense its environment. In some groups, like cephalopods, squid, octopus, the mantle is highly muscular. Its powerful rhythmic contractions are used for jet propulsion, forcefully expelling water from the mantle cavity to achieve rapid movement, for escape or predation.
Ah, so the squid's jet propulsion comes from the mantle muscles.
Precisely. Also, the mantle cavity, created by the mantle, as we said, not only houses gills, but also facilitates water circulation crucial for respiration, feeding in filter feeders like bivalves, and waste removal, receiving outputs from the anus and nephridia. Pigment cells, chromatophores, embedded in the mantle tissue allow for dramatic color changes in cephalopods for camouflage or communication. So the mantle is a truly versatile organ involved in shell formation, respiration, sensation, locomotion, and even defense or display. Its adaptability has been key to molluscan diversification.
Very versatile indeed. Okay, next phylum. Echinodermata, spiny skin.
Yes, phylum Echinodermata. The name literally means spiny-bodied, from Greek echinos, spiny derma, skin, referring to their distinctive endoskeleton. This internal skeleton is composed of numerous small hardened plates or spines called calcareous ossicles embedded within their skin, giving them a rough or spiny texture. A key point: all echinoderms are exclusively marine animals, so you won't find them in freshwater or on land. Organizationally, they are at the organ system level, are triploblastic and coelomate animals, possessing a true coelom, which is extensive.
And they have that weird symmetry thing, right?
They do. It's a unique and frequently tested feature, their symmetry paradox. Adult echinoderms exhibit radial symmetry, typically pentaradial, a 5-part symmetry like a starfish. However, their larval stages are distinctly bilaterally symmetrical.
So, like we discussed before, they probably evolved from bilateral ancestors.
Exactly. The radial symmetry in adults is considered a secondary adaptation, likely linked to their often sessile or slow-moving, bottom-dwelling lifestyle. Their digestive system is usually complete, often with the mouth located on the lower oral or ventral side, and the anus on the upper aboral or dorsal side.
But what's their most distinctive feature?
Arguably, their most distinctive and unique feature, essential for their survival and often asked about in exams, is the water vascular system.
Ah, yes, the internal plumbing.
Precisely. This is a complex hydraulic system derived from the coelom. It consists of a network of interconnected, fluid-filled canals containing seawater and numerous small external projections called tube feet, podia. This system is crucial for several functions, primarily locomotion, allowing them to crawl slowly but powerfully, capture and transport of food, tube feet can manipulate prey, and it also plays a significant role in respiration, gas exchange across the thin walls of the tube feet. Curiously, a specialized excretory system is absent in echinoderms. Waste removal largely occurs by diffusion across body surfaces, including the tube feet and specialized gill-like structures called papillae. Sexes are usually separate, dioecious, reproduction is sexual, and fertilization is typically external, occurring in the seawater. Development is indirect, involving free-swimming, bilaterally symmetrical larval stages like the Bipinnaria or Pluteus larva.
Key examples to remember.
Definitely Asterias, the common starfish, the sea star, Echinus, sea urchin, Antodon, sea lily or feather star, Cucumaria, sea cucumber, and Ophiura, brittle star.
That water vascular system is truly remarkable. For our students who really need to visualize these complex processes for exams, could you elaborate a bit more on how it actually functions? Maybe walk us through how the tube feet extend and retract using that hydraulic pressure. Make it memorable.
Absolutely. Let's visualize the water vascular system in action, step by step. It's fundamental to understanding how echinoderms operate. Imagine it starts with a sieve-like plate on the arboreal upper surface called the madreporate. Think of it as the water inlet. Seawater enters through the madreporate, gets filtered, and flows down a tube called the stone canal. The stone canal connects to a circular canal around the mouth area, the wing canal. Radiating outwards from this ring canal, like spokes on a wheel, are several radial canals, typically one extending into each arm in a starfish.
Okay, madreporate, stone canal, ring canal, radial canals, like a central plumbing system.
Exactly. Now, branching off along each radial canal are hundreds of small paired structures, the tube feet, podia. Each tube foot typically consists of two parts. An internal bulb-like muscular sac called the ampulla, which lies within the coelom, and an external slender projection, the podium itself, which often ends in a suction cup.
Ampulla inside, podium with sucker outside. Got it. How do they move?
Here's the hydraulic magic. 1. Extension. To extend a tube foot, the ampulla, the internal bulb, contracts. This forces the fluid contained within it down into the podium, the external part. The podium elongates and extends outwards due to this hydraulic pressure. 2. Attachment. Once the extended podium touches a surface, muscles in its tip can create a slight suction, allowing it to adhere firmly, like a tiny suction cup. 3. Retraction and movement. To retract the foot, longitudinal muscles within the podium contract. This forces the fluid back up into the ampulla, causing the ampulla to bulge and the podium to shorten and detach its suction.
So squeezing the bulb pushes the foot out, pulling the foot in fills the bulb back up.
Precisely. Now, imagine thousands of these tube feet working in a coordinated fashion. Waves of extension, attachment, and retraction pass along the arms. This allows the echinoderm to crawl slowly, but with incredible force enough for a starfish to gradually pry open the shells of a clam or mussel. This same system is used to grasp food and bring it towards the mouth, and the thin walls of the tube feet also allow for gas exchange. It's truly a marvel of invertebrate hydraulic engineering.
Wow. Hydraulics in action. Very clear now. Okay, our next phylum is Phylum Hemichordata. These used to be chordates.
Yes, Phylum Hemichordata. This is an interesting one because, as you say, Hemichordata was previously considered a subphylum under Chordata. However, it is now recognized as a separate phylum under the non-chordates. This reclassification itself is an important point for your exams.
Why were they initially grouped with chordates?
The main reason for their historical classification under Chordata was the presence of a rudimentary rod-like structure found in their collar region called the stomachoid. This structure arises as an outgrowth of the gut and bears some resemblance to the notochord.
Stomachoid, not a chord. Okay. So what are they like?
Hemichordates are a small group of exclusively marine worm-like animals. They exhibit the organ system level of organization. They are bilaterally symmetrical, triploblastic, and coelomate animals. Their soft, fragile body is typically cylindrical and divided into 3 distinct regions. An anterior muscular proboscis, used for burrowing and feeding, a short, fleshy collar behind it, and a long trunk. Their circulatory system is of the open type. Respiration typically occurs through paired pharyngeal gill slits, openings in the pharynx wall, which is a feature shared with chordates. Their primary excretory organ is considered to be the proboscis gland, located within the proboscis. Sexes are separate, dioecious. Fertilization is external, occurring in the water. Development is usually indirect, involving a free-swimming Tornaria larva, which interestingly resembles the larva of echinoderms.
That larval similarity is interesting too. Examples.
The main examples to remember are Balanoglossus, commonly called the acorn worm, and Saccoglossus.
The reclassification of Hemichordata is an interesting point, showing how science evolves. What specific evidence really led scientists to decide it should be a separate phylum rather than a subphylum of Chordata? And why is that distinction important for students preparing for exams?
That reclassification is indeed a perfect example of the scientific process, and understanding the why can definitely help answer assertion reasoning type questions. The primary reason Hemichordata was moved out of Chordata was the detailed reevaluation of the stomach cord. While initially thought to be homologous, having a shared evolutionary origin, to the chordate notochord based on its position and rod-like appearance, more thorough embryological and molecular studies revealed crucial differences. The stomach cord develops differently. It's an anterior outpocketing of the foregut, essentially part of the digestive tract. It lacks the specific structural composition, like the sheath layers, and the crucial developmental signaling functions of a true notochord. So it's now considered an analogous structure, similar function appearance but different origin, not homologous.
So the stomach cord isn't a real notochord. What else?
Furthermore, hemichordates lack the other definitive chordate characteristics. They do not possess a dorsal hollow nerve cord. Their main nerve cord is typically ventral and solid, though there might be a short dorsal hollow section in the collar. Its homology is debated. They also lack a true post-anal tail. They do have pharyngeal gill slits, which is a chordate feature, but having only one out of the 4 key chordate characteristics isn't enough to classify them within Chordata.
Makes sense. You need the whole package.
Exactly. This distinction is important for students because it reinforces the precise definition of phylum Chordata, stressing that the presence of all 4 fundamental characteristics, notator cord, dorsal hollow nerve cord, pharyngeal gill slits, post-anal tail, at some stage of life is required. It emphasizes that superficial similarities or sharing just one feature aren't sufficient for classification if underlying homology and the complete set of defining traits are absent. It clarifies the boundary between chordates and non-chordates.
Very clear. Hemichordates are out, chordates are next.
Finally, yes, we arrive at phylum Chordata. This is the phylum we belong to. Animals belonging to phylum Chordata are fundamentally characterized by the presence of 4 key diagnostic features, all of which are absolutely essential to know for NEET. 1. Presence of a notochord at some stage in their life cycle. Remember that flexible rod providing support. 2. Presence of a dorsal hollow nerve cord. Unlike the typically ventral and solid nerve cord of non-chordates, the chordate nerve cord runs along the back and is tubular. It develops into the brain and spinal cord in vertebrates. 3. Presence of paired pharyngeal gill slits. These are openings that connect the pharynx, the part of the throat behind the mouth, to the outside. In aquatic chordates, they function in respiration, but in terrestrial chordates, they're usually present only in the embryonic stage. 4. Presence of a post-anal tail. This is a muscular tail that extends posterior to the anus. It may be present only embryonically in some groups.
Notochord, dorsal hollow nerve cord, gill slits, post-anal tail, the 4 must-haves.
Absolutely. Chordates are also bilaterally symmetrical, triploblastic, and coelomate, exhibiting the highest organ system level of organization. They also consistently possess a closed circulatory system. Now, it's crucial to be able to clearly distinguish chordates from non-chordates based on these features. Let's recap the key differences, often presented in tables in your textbooks. Notochord, present in chordates, absent in non-chordates, central nervous system, CNS, dorsal, hollow, and single in chordates, typically ventral, solid, and often double in non-chordates if a nerve cord is present. Pharynx, perforated by gill slits in chordates. Gill slits are absent in non-chordates. Heart, ventral, located on the front belly side in chordates. Dorsal on the back side, if present in non-chordates. Post-anal tail, present in chordates, at least embryonically, absent in non-chordates.
Those comparisons are vital for objective questions. Okay, how is chordate itself divided?
Phylum Chordata is broadly divided into 3 subphyla. 1. Urochordata, also called tunicata. In these marine animals, the notochord is present only in the tail of the larva and disappears in the adult stage. Examples include Ascidia, Salpa, Doliolum. They often have a tunic-like outer covering as adults. 2. Cephalochordata. In this group, the notochord extends from the head region right to the tail region, and importantly, it is persistent throughout their entire life. They are also exclusively marine. The classic example is Branchiostoma, commonly known as Amphioxus or lancelet.
Urochordata and Cephalochordata, are these the protochordates?
Yes, exactly. Urochordata and Cephalochordata are often collectively referred to as protochordates. They represent the earliest, more primitive chordates. They are chordates, but they lack a vertebral column. And that brings us to the third subphylum, where things get really interesting and complex for your exam perspective, Vertebrata. Members of subphylum Vertebrata possess a notochord during their embryonic period, but in the adults, this notochord is replaced by a more robust, segmented structure, the cartilaginous or bony vertebral column, the backbone.
So the backbone replaces the notochord. This leads to that famous statement.
Precisely. This leads us to that crucial statement, a very common assertion reasoning type question in NEET. All vertebrates are chordates, but all chordates are not vertebrates. Let's break that down. It's true because all vertebrates had a notochord during their embryonic development, fulfilling the chordate requirement, which is later replaced by the vertebral column. So they are definitely chordates. However, the protochordates, Urochordata and Cephalochordata, are chordates. They have a notochord, but they are not vertebrates because their notochord is not replaced by a vertebral column. They lack a backbone. This distinction is absolutely fundamental.
Got it. Vertebrates are chordates with a backbone replacing the notochord. What else defines vertebrates?
Besides the basic chordate characters and the vertebral column, vertebrates typically have a ventral muscular heart with 2, 3, or 4 chambers, kidneys for excretion and osmoregulation, and usually paired appendages, which may be fins, like in fish, or limbs, like in tetrapods.
Okay. And how is vertebrata further divided?
Vertebrata itself is divided based on the presence or absence of jaws. Agnatha lacks jaws. This division includes only one living class Cyclostomata. Nithostomata bears jaws. This is the vast majority of vertebrates. It's further divided into 2 superclasses. Superclass Pisces, which bear fins. This includes the fishes. Class Chondrixes, cartilaginous fishes. And class Ostectili, or bony fishes. Superclass Tetrapoda, which bear limbs. This includes 4 classes. Class Amphibia, Class Reptilia, Class Aves, birds, and Class Mammalia.
Agnatha versus Nithostomata, then Pisces versus Tetrapoda. Okay, a clear hierarchy. That distinction, all vertebrates are chordates, but all chordates are not vertebrates, is definitely a classic and often catches students out. You explained the difference, but can you elaborate on the evolutionary advantage of replacing the notochord with a vertebral column? Why was that transition so beneficial for the progression of vertebrate life?
That's a fantastic question because it gets to the heart of why vertebrates became so successful. Replacing the relatively simple, flexible notochord with a segmented, typically bony or cartilaginous vertebral column provided several profound evolutionary advantages. Firstly, the vertebral column offers significantly stronger and more robust axial support compared to the notochord. This increased structural rigidity allowed for the evolution of much larger body sizes. Being bigger has many advantages: better predator deterrence, ability to capture larger prey, greater capacity for internal complexity.
Stronger support allows for bigger bodies. Makes sense.
Secondly, the segmented nature of the vertebral column, composed of individual vertebrae, provides greater flexibility combined with strength. It allows for controlled bending and twisting movements. Crucially, it also provides numerous points for muscle attachment. Muscles attaching to individual vertebrae enable more powerful, precise, and complex movements, leading to more sophisticated locomotion on land, in water, and eventually in air. Yes. Thirdly, the vertebral column provides vastly superior protection for the central nervous system. The delicate dorsal hollow nerve cord, spinal cord, runs through arches in the vertebrae, encasing it in bone or cartilage. This protection is vital for an active organism with a complex nervous system. Finally, especially in bony vertebrates, the vertebral column serves as a significant reservoir for calcium and phosphate, minerals essential for various physiological processes. In essence, the vertebral column transformed a simple supporting rod into a dynamic, protective, and highly adaptable internal skeleton. It was a key innovation that literally formed the backbone for the incredible diversification and ecological success of vertebrates across virtually all habitats.
A truly pivotal evolutionary upgrade.
Yeah.
Okay, let's delve into the first class under vertebrata. Class Cyclostomata, the jawless ones.
Right, class Cyclostomata. These are fascinating because they are the only living representatives of the agnathid division, the jawless vertebrates. All living cyclostomes are ectoparasites on some species of fishes. They attach to the host fish using their specialized mouth and feed on blood or body fluids. They have an elongated eel-like body. Respiration occurs through 6 to 15 pairs of gill slits. Their mouth is distinctively sucking and circular, and crucially, it is without jaws.
No jaws, circular mouth. What about fins and scales?
Their bodies are entirely devoid of scales, having smooth skin instead. They also lack paired fins, like pectoral or pelvic fins. Their internal skeleton, including both the cranium, skull, and the rudimentary vertebral column, is entirely cartilaginous, not bony. They possess a closed type of circulatory system with a two-chambered heart.
And their life cycle. You mentioned migration.
Yes, their life cycle is quite interesting. Most cyclostomes are marine, but they undertake a significant migration into fresh water for spawning, laying eggs. After spawning, the adults typically die within a few days. Their larvae, called Amacoetes larvae in lampreys, live in fresh water for several years, then undergo metamorphosis and migrate back to the ocean to mature.
Quite a journey. Key examples.
The two main examples to remember are Petromyzon, the lamprey, and Myxeme, the hagfish. Hagfish are slightly different. They're mostly scavengers rather than parasites and don't migrate in the same way, but they are still jawless cyclostomes.
Okay, lamprey and hagfish.
Yeah.
Jawless, parasitic, scavenging, cartilaginous. No scales or paired fins.
Got it. Now moving into the jawed vertebrates, Gnathostomata and the superclass Pisces. First up is class Chondrichthyes, cartilaginous fishes. Exactly. Class Chondrichthyes. This group includes sharks, rays, skates, and chimeras. They are predominantly marine animals, known for their streamlined bodies adapted for swimming. Their defining feature is having an endoskeleton made entirely of cartilage, not bone, though it can be calcified for strength. Their mouth is typically located on the lower or ventral side of the head. Unlike higher vertebrates, the notochord is persistent throughout life, although it may be constricted by the developing cartilaginous vertebrae. They have 5 to 7 pairs of gill slits for respiration, which are visible externally as separate openings because, importantly for exams, they lack an operculum, the protective bony gill cover found in bony fishes.
No operculum, that's a key difference. What about their skin and teeth?
Their skin is tough and covered with characteristic minute tooth-like scales called placoid scales. These scales have the same structure as vertebrate teeth, pulp cavity, dentine, enamel layer, and give the skin a rough, sandpaper-like texture. In fact, their teeth are simply modified placoid scales. They are typically numerous, sharp, often backwardly directed, and arranged in rows in the jaws. Tooth replacement is continuous. This makes their jaws very powerful, and most are effective predators.
And that famous point about buoyancy.
Yes, a very important point for competitive exams is the absence of an air bladder or swim bladder in most chondrichthyes.
Right.
This gas-filled sac helps bony fish control buoyancy. Since cartilaginous fish lack it, they are denser than water and must swim constantly using their fins for lift to avoid sinking.
Constant swimming required. What about their heart and temperature?
Their heart is two-chambered, consisting of one auricle and one ventricle, pumping deoxygenated blood to the gills. They are cold-blooded, porcilothermous animals, meaning their internal body temperature varies with the surrounding environment. Some possess interesting special features. For example, some rays have electric organs, torpedo, capable of delivering powerful shocks, while others possess a venomous poison sting on their tail, trigon.
Electric rays and stingrays. How about reproduction?
Sexes are separate, dioecious. In males, the inner edges of the pelvic fins bear rod-like structures called claspers, which are used for transferring sperm during copulation. Fertilization is therefore internal. Many species are viviparous, giving birth to live young, while others are ovoviparous, eggs hatch internally, or oviparous, lay large, yolky eggs, often in protective cases called mermaid's purses.
Internal fertilization and live birth are quite advanced for fish. Examples.
Prominent examples include Scoliodon, the dogfish, often dissected, Pristis, the sawfish with its long-toothed rostrum, Carcharodon, the great white shark, and Trigon, the stingray.
The absence of an air bladder in cartilaginous fishes is indeed a key distinguishing feature for exams.
Yeah.
How does this characteristic specifically relate to their often predatory lifestyle? And what other unique adaptations, besides the teeth and scales, make these powerful swimmers such effective hunters in the marine environment?
That's a great link to make. The absence of an air bladder, while seeming like a disadvantage for buoyancy, is actually quite well suited to their active, often predatory lifestyle. Since they lack this passive buoyancy control, they must swim continuously to generate lift, using their pectoral fins and the shape of their body, and to avoid sinking. This enforced perpetual motion keeps them constantly cruising through the water column, actively searching for prey. It's a lifestyle built around movement. This continuous swimming also naturally facilitates ram ventilation, forcing water over their operculum-less gills for respiration.
So being forced to swim keeps them hunting.
In a way, yes. It fits the profile of an active predator. Beyond this, their success as hunters stems from several other key adaptations. We mentioned the ventrally located mouth and powerful jaws armed with those sharp, replaceable placoid teeth for delivering devastating bites. Many sharks possess an incredibly acute sense of smell, olfaction, capable of detecting minute concentrations of blood or other chemicals in the water from great distances. They also have the lateral line system, common to many fish, which detects water movements and vibrations, helping them locate struggling prey or navigate. Unique to chondrichthyes are the ampullae of Lorenzini. These are specialized electroreceptor organs concentrated on the head, which can detect the very weak electrical fields generated by the muscle contractions of hidden or buried prey. It's like a sixth sense for hunting.
Electroreception. Amazing.
Yes. Combined with their streamlined body shape, powerful heterocircle tail fin, upper lobe larger than the lower, providing thrust and lift, and often counter-shaded coloration for camouflage, these adaptations make them formidable marine predators. Internal fertilization and often giving birth to live, well-developed young, viviparity, also enhances offspring survival rates compared to laying vast numbers of vulnerable eggs.
A whole suite of predatory adaptations. Very impressive.
Okay.
Okay, now for the other major fish class. Class Osteichthyes, the bony fishes.
Yes, class Osteichthyes. This is the largest class of vertebrates, encompassing the vast majority of fish species we know. It includes both marine and freshwater fishes. Their defining feature is having an endoskeleton made primarily of bone, unlike the cartilage of chondrichthyes. Their bodies are typically streamlined, though shapes vary greatly. Their mouth is usually located at the anterior tip or terminal position, though variations exist. They typically possess 4 pairs of gills for respiration, and these gills are covered and protected by a bony flap on each side called the operculum.
Ah, the operculum. That's a key difference from sharks and rays.
A major difference, yes. Allows them to breathe while stationary. Their skin is typically covered with overlapping scales, but these are usually of the cycloid, smooth-edged, or keptenoid, comb-edged type, which are structurally different from the placoid scales of cartilaginous fish. Some bony fish lack scale.
And the buoyancy issue.
Here's the other crucial contrast with chondrichthyes. Most bony fishes possess an air bladder, or swim bladder. This is a gas-filled sac, usually connected to the pharynx, that allows them to precisely regulate their buoyancy and remain suspended at a specific depth without continuous swimming. A huge energy saver.
Operculum and air bladder, 2 big advantages. Heart. Temperature.
Their heart is also 2-chambered, 1 auricle, 1 ventricle, similar to cartilaginous fish, and they are also generally cold-blooded poikilothermous animals. Reproduction varies, but typically sexes are separate, dioecious. Fertilization is usually external, with females releasing large numbers of eggs into the water and males releasing sperm over them. They are mostly oviparous, egg-laying, and development is usually direct. No distinct larval stage. Young resemble small adults.
Examples are numerous here too, right?
Absolutely. Let's categorize some common ones for exams. Marine examples. Exocatus, flying fish glides using enlarged pectoral fins. Hippocampus, seahorse unique posture and male brooding. Freshwater examples: labeo, rohu, important food fish in India. Catla, another major carp. Clarius, magur walking catfish can breathe air. Aquarium examples: betta, Siamese fighting fish. Pterophyllum, angelfish.
The operculum and air bladder are indeed key differences from chondrichthyes and seem like significant advantages. Could you specifically outline how these 2 features function to make bony fish, perhaps, more efficient or adaptable in their aquatic habitat compared to cartilaginous fish? This seems like a practical difference, important for understanding their ecological success.
Absolutely. The operculum and the air bladder are critical adaptations that provide significant functional advantages, contributing greatly to the ecological success and diversity of bony fish. Let's take the operculum first. This protective bony flap covering the gills isn't just passive protection. It plays an active role in breathing through a mechanism called buccal pumping. By coordinating movements of the mouth floor and the opercula, bony fish can actively draw water into the mouth and then force it over the gills and out from under the operculum, even when they are completely stationary.
So they don't have to swim to breathe, unlike many sharks.
Exactly. Cartilaginous fish often rely heavily on ram ventilation, swimming forward with the mouth open. Bony fish can use ram ventilation when swimming fast, but the ability to breathe efficiently while stationary or moving slowly via buccal pumping is a huge advantage. It conserves significant energy, allows them to live in habitats with low water flow, like ponds or deep water, lets them hide among weeds or rocks waiting for prey, or rest on the bottom without suffocating.
Big energy saver and opens up new lifestyles. What about the air bladder?
The air bladder's primary role is providing neutral buoyancy. By precisely regulating the amount of gas, usually oxygen, nitrogen, CO2 extracted from the blood, within the bladder, bony fish can adjust their overall density to match that of the surrounding water. This means they can remain suspended at any desired depth in the water column without expending muscular energy to counteract sinking, like chondrichthyes must do, or floating up.
Another huge energy saver.
Tremendous. This conserved energy can be allocated to other activities like foraging, growth, escaping predators, or reproduction. It also gives them much finer control over their vertical position in the water column, allowing them to effectively exploit different depth and niches within their aquatic environment. Some fish even use the air bladder for sound production or reception. So combined, the operculum allows for energy-efficient respiration in diverse conditions, and the air bladder provides energy-efficient buoyancy control. These two features significantly reduce the energy costs of living underwater, contributing immensely to the ecological dominance and diversification of Osteichthyes.
Brilliant adaptations. Okay, moving out of the water now, mostly. Class Amphibia.
Yes, class Amphibia. The name itself gives the clue, from Greek ampha meaning dual and bios meaning life. This signifies their characteristic ability to live a dual life, successfully inhabiting both aquatic, especially for reproduction in larval stages, and terrestrial habitats as adults. This group includes frogs, toads, salamanders, newts, and caecilians. Most amphibians possess two pairs of limbs, they are tetrapods, adapted for walking, jumping, or swimming. Their body is typically divisible into a distinct head and trunk. A tail may be present in some adults, like salamanders, but is absent in others, like frogs and toads.
And their skin is very different from fish.
Yeah, very different. A key characteristic is their smooth, moist skin, which is typically without scales, though tiny embedded scales exist in some caecilians. This moist skin is rich in mucus glands, keeping it damp, and crucially, it plays a significant role in respiration, cutaneous respiration. Their eyes usually have movable eyelids. Unlike fish, they possess a middle ear cavity and an eardrum, the tympanum, which is visible externally on the side of the head and represents the ear. A feature shared with reptiles and birds is the cloaca. This is a common chamber at the posterior end into which the alimentary canal, digestive tract, urinary ducts, and reproductive tracts all open. Waste products and gametes exit the body through this single cloacal opening.
Okay, cloaca is the common exit. How do they breathe? You mentioned the skin.
Amphibians exhibit a fascinating diversity of respiratory methods throughout their lives. Larval stages, like tadpoles, typically breathe using external or internal gills. Adult amphibians primarily breathe using lungs, which are relatively simple sacs. However, respiration through their moist skin, cutaneous respiration, is also very important, sometimes accounting for a significant portion of gas exchange, especially when underwater or hibernating. Some amphibians even have gas exchange across the lining of their mouth cavity. Buccopharyngeal respiration.
Gills, lungs, skin, very adaptable. Heart, temperature.
Their heart is three-chambered, consisting of two auricles and one ventricle. This allows for some separation of oxygenated and deoxygenated blood, but mixing does occur in the single ventricle. They are cold-blooded poikilotherms, relying on external sources to regulate their body temperature.
And reproduction. Back to the water.
Yes, reproduction typically ties them to water. Sexes are separate, dioecious. Fertilization is usually external in most frogs and toads, occurring in water as the female lays eggs and the male releases sperm over them. Salamanders often have internal fertilization via a sperm packet, spermatophore. They are generally oviparous, laying shellless eggs, usually in water or very moist environments, as the eggs lack protection against drying out. Development is indirect, involving an aquatic larval stage, like the tadpole and frogs, that undergoes a dramatic metamorphosis to transform into the terrestrial or semi-aquatic adult form.
Metamorphosis is key. Examples.
Common examples include bufo, toad, rana, frog like the Indian bullfrog, Hoplobrachus tigerinus, Hyla, tree frog, salamander, and Ichthyophis, a limbless amphibian resembling a worm found in India.
Amphibians are truly fascinating as these transitional forms between water and land. What specific adaptations really allow them to survive successfully in both environments? And how crucial are these adaptations for their survival, especially thinking about changing or fluctuating environments like we see with climate shifts today?
Their success, and indeed their vulnerability, hinges on a suite of adaptations that bridge the aquatic and terrestrial worlds. It's a delicate balancing act. Firstly, their dual respiratory mechanisms are absolutely key. Larval gills are perfect for underwater life. Adult lungs allow air breathing on land, but the critical supplement is their moist, permeable skin. This allows for significant cutaneous respiration, enabling them to absorb oxygen directly from water or even moist air when submerged or inactive. This skin is also important for water absorption.
But that permeable skin must be a disadvantage too.
It is, absolutely. It's a double-edged sword. While it aids gas and water exchange, it makes them highly susceptible to water loss, desiccation, in dry conditions. This fundamentally ties most amphibians to moist habitats or forces them into inactivity during dry periods. It also makes them incredibly vulnerable to absorbing pollutants and pathogens from their environment, which is why they are often considered excellent bioindicators of environmental health. Secondly, the evolution of four limbs, tetrapod structure, was obviously a major adaptation for locomotion on land, allowing them to move away from the water's edge, although their limb structure is less efficient for sustained running than later tetrapods. Thirdly, their reproductive strategy inherently links them to water. Laying shellless eggs that require a moist environment and having an aquatic larval stage, like tadpoles, means they must return to water or very damp places to breed, even if the adults are primarily terrestrial.
So they can live on land, but need water to reproduce and keep their skin moist.
Exactly. From an evolutionary perspective, these adaptations represent a crucial bridge, the first vertebrates to truly venture onto land, but they never fully severed their ties to the aquatic environment. In the context of climate change, this dependence makes them extremely vulnerable. Fluctuations in rainfall patterns directly threaten their breeding sites, ponds, streams drying up. Increased temperatures can accelerate desiccation, restricting their activity periods and habitable ranges. Habitat destruction and pollution, easily absorbed through their skin, add further pressure. This combination of factors is why amphibians are currently experiencing one of class name highest rates of decline and extinction among all vertebrate groups globally. Their transitional nature makes them particularly sensitive to environmental shifts.
A critical point about their vulnerability. Okay, let's move on to the next class, the ones that truly conquered land. Class Reptilia.
Yes, class Reptilia. This group includes lizards, snakes, turtles, tortoises, crocodiles, and alligators. The class name comes from the Latin repere or reptum, meaning to creep or crawl, referring to their typical mode of locomotion, though some run, swim, or climb very effectively. Reptiles are primarily terrestrial animals, although many have adapted to aquatic or semi-aquatic life. Their major evolutionary success on land is largely due to adaptations that reduce water loss. The most significant is their skin. Their body is covered by dry and cornified skin, featuring epidermal scales or scutes, bony plates in some, like turtles and crocodiles. This tough, keratinized outer layer acts as a waterproof barrier, critically helping to prevent water loss by evaporation, enabling them to thrive even in arid environments where amphibians cannot survive.
So dry, scaly skin is the key difference from amphibians. What about ears and limbs?
They generally do not have external ear openings. Pinnae. Like amphibians, a tympanum, eardrum, often recessed, represents the ear. Limbs, when present, are usually two pairs, typically with 5 clawed digits, adapted for running, climbing, or paddling. However, limbs are completely absent in snakes and some lizards.
Arch chambers, still 3.
Usually 3-chambered, 2 auricles, and 1 partially divided ventricle, allowing for slightly better separation of oxygenated and deoxygenated blood than in amphibians. However, crocodiles are an important exception. They possess a fully 4-chambered heart, similar to birds and mammals, allowing for complete separation. This is a key point to remember for exams. Like amphibians and fish, reptiles are poikilotherms, cold-blooded, meaning they rely on external heat sources, like basking in the sun, to regulate their body temperature.
And they shed their skin.
Yes, many reptiles, particularly snakes and lizards, periodically shed the outer layer of their skin all at once or in patches. This process is called extatesis, or sloughing. Reproduction is also adapted for land. Sexes are separate, dioecious. Fertilization is internal. A major advancement is that they are primarily oviparous, laying amniotic eggs.
Amniotic eggs, that's a big deal, right?
Huge! The amniotic egg has a protective shell, leathery or calcareous, and internal membranes, amnion, chorion, allantois, yolk sac, that protect the embryo from drying out, provide nutrients, store waste, and facilitate gas exchange. This allowed reptiles to reproduce entirely on land, completely breaking the dependence on water for reproduction that amphibians have. Development is direct the young hatch as miniature versions of the adult, no larval stage.
Amniotic egg equals true independence from water for reproduction. Examples.
Many diverse examples. Chelone, marine turtle, testudo, tortoise, chameleon, tree lizard, coelomates, garden lizard, hemidactylus, wall lizard, crocodilus, crocodile, alligator, and the snakes, naja cobra, bangerous crate, vipera, viper note, which are poisonous, also non-poisonous snakes like python.
The dry, cornified skin is clearly a major adaptation allowing reptiles to thrive on land. Comparing it directly to the moist skin of amphibians, how significant was this change in terms of evolutionary advancement, especially regarding water conservation and their overall success in terrestrial environments?
The transition from the moist, permeable skin of amphibians to the dry, cornified, scaly skin of reptiles marks a monumental evolutionary advancement. It was arguably one of the most critical steps enabling vertebrates to truly conquer terrestrial environments. Think back to the amphibian skin. Its permeability is essential for cutaneous respiration and water absorption, but it's a massive liability in dry air, leading to rapid desiccation. This fundamentally restricts most amphibians to moist habitats and limits their size and activity patterns on land. In stark contrast, the reptilian skin, with its thick outer layer of dead, keratin-filled cells forming scales or scutes, acts as an incredibly effective waterproof barrier. Keratin is a tough, fibrous protein that is largely impermeable to water. This drastically reduces evaporative water loss from the body surface.
So they could move away from water sources without drying out.
Precisely. This single adaptation allowed reptiles to largely break their physiological dependence on water or high humidity for survival. It enabled them to colonize a much wider range of terrestrial habitats, including hot, arid environments like deserts and grasslands, completely inaccessible to most amphibians. This skin also provides superior physical protection against abrasion and injury compared to the delicate skin of amphibians. With reduced reliance on the skin for gas exchange, reptiles evolved more complex and efficient lungs, further supporting a more active terrestrial lifestyle. So, in terms of evolutionary significance, the development of dry, cornified skin, along with the amniotic egg, represents the key innovations that led to reptiles' greater independence from water. This allowed for their widespread radiation and diversification across land, paving the way for the subsequent evolution of other fully terrestrial amniotes, birds, and mammals.
A game-changing adaptation. Okay, taking to the skies next. Class Aves, the birds.
Yes, class Aves, the birds. Their most characteristic and obvious features are the presence of feathers, unique structures derived from scales, and, for most species, the ability to fly, though some, like ostriches, emus, kiwis, penguins, are flightless. Birds possess a beak or bill, which is essentially modified jaws covered in keratin. Importantly, modern birds lack teeth. Their forelimbs are highly modified into wings, the primary structures for flight. Their hind limbs are typically adapted for walking, running, perching, often having scales like reptiles, or swimming.
Feathers, wings, beak, no teeth. What about their skin and skeleton?
Their skin is generally dry and thin, lacking glands, with one notable exception. The oil gland, or preen gland, uripidal gland, located at the base of the tail. Birds use the oily secretion from this gland to waterproof and maintain their feathers. Their endoskeleton is fully ossified, bony, but remarkably adapted for flight by being both strong and lightweight. Many long bones are hollow and contain air cavities, pneumatic bones, connected to the respiratory system. This significantly reduces the overall weight of the skeleton. The sternum, breastbone, is often large and keeled, providing a large surface area for the attachment of powerful flight muscles.
Pneumatic bones for lightness.
Okay.
Little digestive system.
Their digestive system is adapted for rapid processing of food to meet high-energy demands. The digestive tract often has additional chambers. The crop, an expansion of the esophagus used for temporary food storage, and the gizzard, a muscular posterior part of the stomach often containing small stones, used for grinding hard food, compensating for the lack of teeth.
Crop and gizzard, heart and temperature, big changes here.
Yes, major advancements. Birds possess a completely four-chambered heart, 2 auricles, 2 ventricles, ensuring complete separation of oxygenated and deoxygenated blood. This allows for highly efficient oxygen delivery to the tissues. And crucially, birds are warm-blooded homeothermous animals. They can maintain a high, constant internal body temperature independent of the external environment. This high metabolic rate and stable temperature are essential for providing the sustained energy required for flight.
Four-chambered heart, warm-blooded, vital for flight. How do they breathe so efficiently?
Respiration occurs through lungs, but the avian respiratory system is incredibly efficient due to the presence of air sacs. These thin-walled sacs extend into the body cavity and even into the pneumatic bones. They facilitate a unique unidirectional flow of air through the lungs, parabronchi, meaning fresh oxygenated air passes through the lungs during both inhalation and exhalation. This continuous flow maximizes gas exchange efficiency, far exceeding the tidal flow in mammals. This system is vital for meeting the extreme oxygen demands of flight.
Unidirectional airflow via air sacs, super efficient. Reproduction.
Sexes are separate, dioecious. Fertilization is internal. All birds are oviparous, laying hard-shelled amniotic eggs, similar to reptiles but typically with more parental care, incubation, feeding young. Development is direct.
Examples.
Countless familiar examples: corvus, crow; columba, pigeon; psittacula, parrot; struthio, ostrich flightless; pavo, peacock peafowl; aptenidites, penguin flightless, adapted for swimming; neophron, vulture.
Birds exhibit truly remarkable adaptations for flight, beyond the obvious feathers and wings which everyone sees. What are those subtle but really significant internal adaptations like the pneumatic bones and the air sac system you mentioned that truly make sustained flight possible? These seem like key points for exams.
That's a crucial point, because while feathers and wings are the most visible flight hardware, the true marvel of avian flight really lies in these intricate internal modifications. These are frequently tested because they show a deep understanding of adaptation. Firstly, pneumatic bones. We mentioned they're hollow and filled with air spaces connected to the respiratory system. This drastically reduces skeletal weight. A bird's skeleton often weighs less than its feathers. This lightness is fundamental. Reducing weight means less muscle power is needed simply to lift the body, making flight much more energetically feasible. It's a primary weight-saving strategy, absolutely critical for achieving lift and agility in the air.
Lighter bones, easier than liftoff?
Exactly. Secondly, the air sac system, coupled with the relatively rigid lungs, is arguably the most sophisticated respiratory adaptation among vertebrates, perfectly designed for high-energy activities like flight. Remember, it enables unidirectional airflow. Unlike our mammalian lungs, where air flows in and out the same passages, tidal flow, in birds, air flows in one direction through the gas exchange surfaces, parabronchi. The air sacs act like bellows, storing inhaled air and pushing it through the lungs in a continuous stream, even during exhalation.
So the lungs get fresh air constantly, not mixed air like ours.
Precisely. This means the lungs are almost continuously bathed in oxygen-rich air, maximizing the efficiency of oxygen uptake from each breath. This highly efficient gas exchange is absolutely vital to meet the enormous metabolic demands, the incredibly high oxygen consumption rate required for the sustained, powerful muscle activity needed for flapping flight. Combine these with other internal features like the fusion of bones, example, pygostyle in the tail, synsacrum in the pelvis, furcula or wishbone for rigidity and strength, the large keeled sternum for flight muscle attachment, the highly efficient four-chambered heart delivering oxygenated blood effectively, and their homoothermous nature providing constant high body temperature for optimal muscle function. It all forms a perfectly integrated internal system designed for aerial mastery. These less obvious internal features truly underpin the possibility of sustained, powered flight.
Incredible feat of biological engineering. Okay, last but certainly not least, class Mammalia.
Yes, class Mammalia. This is the class we belong to. Mammals are incredibly diverse and are found in almost every habitat on Earth, from polar ice caps to deserts, oceans to forests. Some have even adapted to fly bats or live their entire lives in water, whales, dolphins. The single most unique and defining mammalian characteristic, the one that gives the class its name, is the presence of milk-producing glands, the mammary glands, in females. Milk is used to nourish the newborn young.
Mammary glands equal mammals. What else is unique or characteristic?
Mammals typically possess two pairs of limbs, which are highly adapted for a vast array of functions depending on the species. Walking, running, climbing, burrowing, swimming, or even flying. In bats, the forelimbs are modified into wings. Their skin is also unique in consistently possessing hair or fur at some stage of their life cycle. Hair provides insulation, camouflage, sensory input, whiskers, and protection. External ears, or pinnae, are typically present, helping to collect sound waves. Mammals also generally exhibit heterodont dentition.
Heterodont, meaning different types of teeth.
Exactly. Unlike reptiles or fish, which often have uniform teeth, homodont, mammals typically have different types of teeth incisors, canines, premolars, and molars present in the jaw, each specialized for different functions like cutting, tearing, or grinding food. The specific arrangement, dental formula, varies between groups and is related to diet. Like birds, mammals have a highly efficient four-chambered heart, 2 auricles, 2 ventricles, ensuring complete separation of oxygenated and deoxygenated blood. They are also homeothermous, warm-blooded, capable of maintaining a constant high internal body temperature through metabolic activity regardless of the external temperature. Respiration occurs exclusively by lungs.
Reproduction, similar to birds.
Sexes are separate, dioecious, and fertilization is always internal. The vast majority of mammals are viviparous, meaning they give birth to live young. The embryo develops inside the mother's uterus, receiving nourishment via a placenta in most. However, there are a few exceptions. The monotremes, like the platypus and echidnas found in Australia and New Guinea, are oviparous. They lay leathery shelled eggs like reptiles. This is a key exception to remember. Development in all mammals is direct.
Platypus laying eggs, the famous exception. Examples are everywhere, but maybe some key ones showing diversity.
Sure. Let's list a few representing different groups or adaptations. Oviparous monotreme, Ornithorhynchus, platypus. Viviparous marsupial give birth to underdeveloped young nurtured in a pouch. Macropus, kangaroo, viviparous, placental vast majority. Pteropus, flying fox, bat, flying mammal. Camillus, camel adapted to desert. Macaca, monkey primate. Rattus, rat. Canis, dog. Felis, cat. Elephas, elephant. Equus, horse. Delphinus, common dolphin marine mammal. Balanoptera, blue whale largest mammal. Panthera tigris, tiger. Panthera leo, lion. Homo sapiens, human.
Mammals are often considered to be at the peak of evolutionary complexity, thriving everywhere. How do those two unique features, mammary glands and hair, specifically represent an adaptive advantage? How have they allowed mammals to diversify so incredibly widely across almost all habitats on Earth?
That's an excellent way to frame it, focusing on why these signature traits are so advantageous. Mammary glands and hair are truly distinguishing features, and their evolution has been profoundly significant for mammalian success and diversification. Let's consider mammary glands first. Their evolutionary impact is huge because they provide reliable, high-quality nourishment for the young that is independent of external food availability or seasonality. Milk is a rich source of proteins, fats, carbohydrates, vitamins, minerals, and antibodies, perfectly tailored for the needs of the growing offspring. This allows the mother to provide consistent sustenance, crucial for early growth and brain development, even in environments where food might be scarce or unpredictable for the young to find themselves.
So it ensures the babies get fed reliably, no matter what.
Exactly. It also means young can be born in a relatively underdeveloped state, altricial, yet still survive and thrive under maternal care. This internal food source fosters extended parental care. The prolonged dependency allows for longer periods of learning, the transmission of complex behaviors from parent to offspring, and the development of strong social bonds, which are characteristic of many mammal groups and vital for higher cognitive development. This strong maternal investment significantly increases offspring survival rates compared to just laying eggs and leaving them.
Okay, mammary glands boost survival and allow for learning. What about hair?
Now, hair, forming fur or pelage, its primary and most crucial adaptive advantage is in thermoregulation, enabling mammals to maintain their high, constant body temperature homeothermy efficiently. Hair traps a layer of air close to the skin, acting as an excellent insulator. This significantly reduces heat loss in cold environments, allowing mammals to survive and remain active in climates far too cold for most reptiles or amphibians. Conversely, in hot environments, specialized hair coats and associated mechanisms like sweating, enabled by skin glands, another mammalian feature, can help dissipate heat.
So it's like a built-in thermostat regulator.
Precisely. This ability to maintain a stable internal temperature, fueled by a generally high metabolic rate, supported by the efficient four-chambered heart and lungs, allows mammals to remain active across an extraordinary range of environmental temperatures, from Arctic cold to desert heat, and also during different times of day or night. This physiological independence from ambient temperature opened up vast ecological opportunities unavailable to poikilotherms. Furthermore, hair serves other important functions: camouflage, matching surroundings, sensory input like whiskers or vibrissae, which are highly sensitive tactile hairs, physical protection against abrasion or UV radiation, and even social signaling, ear gamp raising hackles.
So hair for temperature control, sensing, protection.
Exactly. Together, the ability to nurture young internally with milk, ensuring high survival and learning potential, combined with the capacity to physiologically regulate body temperature using hair and high metabolism, allowed mammals to break free from many of the environmental constraints that limit other vertebrate groups. This enabled them to successfully occupy and diversify into an incredibly vast array of ecological niches across almost every habitat on Earth, from the deepest oceans to the highest mountains, leading to their remarkable evolutionary success and complexity.
Mammary glands and hair, truly key mammalian innovations. Okay, we've covered a massive amount of ground. Let's try to consolidate this for exam readiness. Can you quickly review the absolute core concepts?
Definitely. Let's do a quick recap to solidify the key takeaways for your exams. We started by establishing the fundamental features used for classifying the animal kingdom. Remember these pillars? Levels of organization. Cellular, Porifera; tissue, Coelenterata, Panophora; organ, Platyhelminthes; organ system, Aschelminthes to Chordata. Remember the progression in complexity: symmetry, asymmetrical, most Porifera; radial, Coelenterata, Ctenophora; adult Echinodermata; bilateral, Platyhelminthes to Chordata; larval, Echinodermata; link symmetry to lifestyle; cell organization, germ layers; diploblastic, Coelenterata; Ctenophora, ectoderm, endoderm; versus triploblastic, Platyhelminthes to Chordata, ectoderm, mesoderm, endoderm. Mesoderm allows for greater complexity. Coelom, body cavity. Acoelomate, Platyhelminthes, no cavity. Pseudocoelomate, Aschelminthes, false cavity, not lined by mesoderm. Coelida, Annelida to Chordata, true cavity, lined by mesoderm. Remember the key examples for each. Segmentation, metamerism, absent in many, present distinctly in Annelida, Arthropoda, Chordata. Allows for specialization, notochord. Absent, non-chordates Porifera to Hemichordata versus present, chordates at some life stage. The fundamental divide.
Those basic frameworks are crucial.
Absolutely. Then we systematically covered each major phylum, focusing on their distinctive features, the things that make them unique and are crucial for differentiation in MCQs. Porifera, cellular level, water canal system, ostia, spongocoel, osculum, choanocytes, collar cells, spiculus spongin, Coelenterata, scenaria. Tissue level, ctenoblast nematocysts, stinging cells, pulp medusiforms, metagenesis, obelia, gastrovascular cavity, Ctenophora, cone plates, 8 rows for locomotion, bioluminescence, exclusively maroon, Platyhelminthes, dorso-ventrally flattened body, acoelomate, flame cells, excretion, often parasitic, hook suckers, triploblastic, organ level, Aschelminthes, round body and cross-section, pseudocoelomate, complete alimentary canal, muscular pharynx, sexual dimorphism often present, organ system level, Annelida, largest phylum, chitinous exoskeleton, molting, jointed appendages, defining feature, tagmatization, head, thorax, abdomen, diverse respiratory organs, gills, book lungs, trachea, Malpighian tubules, excretion in insects, open circulation, Mollusca. Second largest, calcareous shell, often present, secreted by mantle. Mantle and mantle cavity, gills, radula, rasping tongue, muscular foot, visceral hump. Echinodermata, exclusively marine, spiny skin, calcareous ossicles, endoskeleton. Water vascular system, tube feet, locomotion, feeding, respiration. Radial symmetry, adults, bilateral symmetry, larvae, no head. Hemichordata, worm-like marine, proboscis, collar, trunk, stomach cord and collar region, gill slits present, open circulation. Chordata, possess all 4 at some stage. Notochord, dorsal hollow nerve cord, pharyngeal gill slits, post-anal tail, closed circulation.
That's a great rapid-fire summary of the phyla. And within Chordata.
Right. Within Chordata, remember the protochordates. Hemichorda has a notochord and larval tail. Cephalochordata notochord persists head to tail. Then the crucial subphylum Vertebrata, defined by the vertebral column replacing the notochord. Remember, all vertebrates are chordates, but not all chordates are vertebrates. And the key distinguishing features of each vertebrate class. Cyclostomata, jawless, sucking circular mouth, no scales or paired fins, cartilaginous skeleton, parasitic scavenging. Lamprey, hagfish. Chondrichthyes, cartilaginous skeleton, placoid scales, no operculum, no air bladder usually. Ventral mouth, internal fertilization, often. Sharks, rays. Osteichthyes, bony skeleton, cycloid, wedgenoid scales, operculum present, air bladder present, terminal mouth usually, external fertilization usually. Vast majority of fish. Amphibia, dual life, waterland, moist skin, no scale, cutaneous respiration, 3-chambered heart, external fertilization usually, metamorphosis, tadpole larva. I need water for reproduction, frogs, salamanders, reptilia, tricorniphyte skin with scalescutes, amniotic egg, kefir land. Internal fertilization, 3-chambered heart, usually 4 in crocodiles. Poikilothermic, lizards, snakes, turtles, crocodiles. Aves, feathers, 4 limbs modified to wings, pneumatic bones, air sacs, unidirectional respiration. 4-chambered heart, homoiothermic, warm-blooded, beak, no teeth, oviparous, hard-shelled eggs, birds, mammalia, mammary glands, milk, hair, pinnae, external ears, heterodont teeth, diaphragm, four-chambered heart, homeothermic, viparous, mostly except monotremes, mammals. I strongly recommend you carefully study Table 4.2 in your NCERT textbook. It provides a fantastic comparative summary of the salient features of all these phyla and classes at a glance. It's an excellent tool for last-minute revision.
We've truly gone on a deep dive today, really unearthing the fascinating world of the animal kingdom and its intricate classification. This systematic understanding, as we've emphasized throughout, is your absolute key to excelling in biology, not just conceptually, but also in securing those vital marks in your board exams and competitive exams like NEET.
Absolutely. Conceptual clarity is paramount. Don't just memorize lists. Understand the why behind the classification, the evolutionary significance of the features. For your upcoming exams, I highly recommend you revise this topic again very carefully. Go back through your textbook, pay close attention to the diagrams. They really help visualize the structures and study those comparative tables. And most importantly, practice past year questions based on this lesson. Focus especially on questions that ask you to distinguish between phyla or classes based on their unique features, or that test your knowledge of specific examples and exceptions. Keep pushing your boundaries. I know this chapter has a lot of detail, but each concept you master, each phylum you understand brings you closer to your goals. Feel free to reach out if you have any persistent doubts about this chapter.
Excellent advice. And one final thought to leave you all with. As you review this incredible, intricate hierarchy of life that we've explored today, consider how the evolution of each fundamental feature, from a simple cellular aggregate to complex organ systems, from asymmetry to bilateral symmetry, from acoelomates to coelomates, the development of segmentation, the notochord, the amniotic egg, how each step allowed animals to conquer increasingly diverse and challenging environments on our planet. Which of these many evolutionary innovations do you personally think was the most pivotal, the biggest game changer for the overall success and diversification of animal life on Earth? Something to ponder.